Hey everybody, welcome to another video lecture. This one is chapter 20, Enzymes and Vitamins. So we're going to start off just by looking at some enzymes, some classification, some terminology, factors that affect how enzymes work, um, how we can turn them off, how we turn them on, some of the things that inhibit them, and then we'll look at some vitamins. Vitamins play an important role because they're often cofactors or coenzymes or some kind of necessary part of enzyme action, or they're often precursor molecules to things that are important for us physiologically. So we'll kind of look at enzymes first and then vitamins. Enzymes are catalysts, and we call them biological catalysts because our bodies make them. They're made out of proteins. And as you would maybe remember from catalysts, catalysts actually make reactions go faster. The way that we say this happens is that there's a lowering of activation energy. And so if you take a look over here at this diagram, this is an activation or an energy diagram where on the left-hand side you see the energy, high to low, low to high, follow the arrow, and then as the reaction starts, you've got your reactants, and they have some initial energy level. And then there's the products, what you're going to get at the end, and this has some usually lower energy level, and high energy going to low energy. This is typically how reactions proceed, and usually these are also spontaneous reactions. They'll happen on their own. Now, there is an energy hump. You can see it right here with the purple line. The molecules have to get into some kind of higher energy state before they'll actually react and then form this lower energy state. And I've got the wrong line there shown. So it's the green one. So high energy state, low energy state. And what an enzyme does is it actually just makes that energy gap a little lower. And it does that in different ways, right? This gap, again, has to be overcome by the molecules getting together really close. Sometimes that's hard to do if they're both, have certain charges that repel. They have to have the right energy, the right orientation. And so what an enzyme does is it actually kind of holds the molecules in just the right way. So it doesn't leave it up to chance. It just holds the molecules together or pulls them apart and actually just lowers the amount of energy required to get to whatever this transitional state is. And so we're not going to look at any specific reactions or specific transition states, but in general, that's what an enzyme does, is it lowers the activation energy. And the activation energy is just that energy hump that has to be overcome whenever a reaction takes place. Catalysts are recyclable. They're able to be used again, and the same is true with enzymes. Enzymes don't get destroyed at the end. Sometimes they have to be, whatever the reaction did to them has to be undone, and then they can go and work again. But usually that takes place at the same time as the reaction's happening. So when the enzyme really kicks out that molecule, those products, it's usually ready to go. So enzymes lower activation energy, and one of the reactions in our bodies that we can look at that has an enzyme that does the work is the carbonic anhydrase reaction. So our body has carbon dioxide and water that get produced by the cells. Now carbon dioxide is a gas, and typically this doesn't dissolve very well in our bodies, and we don't want to have any bubbles in our bodies. And so what carbonic anhydrase does is it converts the CO2 and the H2O into bicarbonate and hydrogen ion. And bicarbonate is soluble, and this helps it get transported to the lungs and all that stuff so that it can be breathed out. It also is part of the physiological buffer that we have in our bodies. If you remember back to just, it was probably the end of Chem 21, or Chem 20, sorry, where you guys would have done acids and bases and maybe talked about buffers. Anyways, this helps to buffer our blood. But this enzyme is responsible for doing a really good job of converting all the CO2 that gets formed into this bicarbonate in a very, very fast manner so that there isn't any bubbles in our blood. So this enzyme is one of those that works really overtime in our bodies. Nearly all enzymes are globular proteins. This means that they are able to dissolve in water, and they're usually, what globular implies, kind of round and clumpy, as opposed to fibrous proteins that make up like your hair and nails and horns and that are more structural and water insoluble. They bind to molecules that are either very specific or that are of the same type, called substrates. Their tertiary structure includes a little pocket called an active site, and this is the site that the substrate actually binds to. Now, there are specific amino acids within that active site that actually bind the substrate, that hold it in place while the enzyme is able to do the catalytic activity to help the reaction in whatever way. Here's an example. Here's lactase. Lactase has an active site. Lactase looks like it's a tetramer, so 4 different protein chains, a yellow, blue, red, and green. You can see there are combinations of alpha helices and beta sheets. Anyways, there's an active site, and in that active site we see lactose, the disaccharide, bound there and held in place by these amino acids. And they're held in place by hydrogen bonding to the R groups, salt bridges to the R groups, hydrophobic, hydrophilic interactions to the R groups. Now, most likely this active site being exposed to, to the cellular solutions and to the place wherever, lactose is floating around, it's probably got polar R groups as opposed to non-polar R groups. And again, to do hydrogen bonding, we would expect those to be some polar R groups. So the substrate is held in place in the active site. Now, as far as the type of binding that an enzyme can do, we can classify it a few different ways. We can say that it shows absolute specificity. Absolute specificity means there's only one type of substrate that it will bind, okay? And it'll do one reaction on that substrate. An example would be urease, and it catalyzes the hydrolysis of urea, and only hydrolysis. There can be group specificity. This means that if you're a similarly acting substance, it'll work on all similarly acting similarly looking substances. So a hexokinase is a 6-carbon sugar, right? Hexoses are 6-carbon sugars. Hexokinase is the name of the enzyme. And a hexokinase will go and it'll add a phosphate group. And just for your knowledge here, kinases add phosphate groups. But a hexokinase specifically will add phosphate groups onto any 6-carbon sugar. It'll react with any hexose. Hexoses, you can imagine, might have different, L or D positions of their OHs, but it doesn't matter. This recognizes the whole group of them. And then there are linkage-specific enzymes, linkage specificity. So it'll actually seek out certain types of bonds, and it doesn't matter what molecule has those bonds, it'll break them. An example of that is chymotrypsin. This is a digestive enzyme. This goes and actually breaks apart peptide bonds. It'll go and chymotrypsin specifically goes and breaks the peptide bond after aromatic amino acids. So any amino acid, something like tryptophan or something like phenylalanine, if they have an aromatic group in them, chymotrypsin recognizes that group and then goes and it cuts a peptide bond, or it breaks the peptide bond. So absolute, group, and linkage are the specificity types. So here's a little bit of the terminology. So when the enzyme is a free enzyme, meaning it hasn't bound to anything, it's just called an enzyme. Likewise with the substrate. When the enzyme and substrate react together and see each other and interact, it's called the ES, or enzyme substrate complex. This is what forms when we say the activation energy gets lowered. And then it instantly turns into an enzyme product complex which will then dissociate. And when I say dissociate, it just means that the enzyme and the product will go their own way. They'll separate. So they form an ES, they form EP, EP dissociates. So that's a little bit of the terminology that you're going to see here, right? Okay. So in this example, we see lactose and the enzyme lactase. So lactose, the free substrate, enzyme lactase here. The active site in this case will recognize structural components of the disaccharide lactose. What's shown here is an active site that doesn't exactly match the shape of the substrate, but we know there will be substrate binding. We know that once it's formed the ES complex, hydrolysis requires a water molecule, so the water's going to come in here. We're going to see, we're not seeing the reaction, but we did see the hydrolysis take place. And then now the EP complex has been formed, and so then it dissociates. We get our monosaccharides, and we get our enzyme again. Now, the way that the substrate binds to the active site, there's a couple of proposed mechanisms. And in some cases, these are both true things. And in some enzymes we see it is very, very lock and key. In others that we see, it's more of an induced fit. So let's talk about them. Lock and key model is a rigid substrate binding. So what that means is that the active site is very much like a lock. The substrate's very much like a key. One has to match it exactly for it to work. And so this is what we see with that absolute specificity, that type of enzyme binding. Absolute meaning it goes for one exact molecule and does one exact reaction. That's because it probably has a lock and key type binding. In the induced fit model, the active site is a little more malleable, and the substrate can actually go in there and sort of push its way around and sort of fit in there. I like to imagine that this is the type of binding that happens with our taste buds, right? We have molecules that will bind to our taste buds and tell us if something is sweet. Same taste bud will get different levels of sweetness depending on what we eat, right? And different things that we eat have different sugars in them. And even sugar substitutes. Sugar substitutes will bind to our taste bud and give us a sensation of sweet. And so what must be happening is that our taste buds are like an induced fit type scenario, and the different shapes of the different sugar molecules and artificial sweeteners bind stronger or weaker, meaning that they can fit in there better or they don't fit in there that well. And that ultimately gives us the strength of the signal that we get to our brain and the overall sensation of the sugar and how sugary it is. So the induced fit model, these things kind of force, I like to think the substrate kind of forces its way in, the enzyme kind of melds around it. There's a lot of evidence to kind of suggest that this might be the case in a lot of situations, because one of the things that happens is once the EP complex forms, right? We've got the ES complex forming, and then it turns into the EP complex. Well, if the EP complex or the ES complex, either one of these, if the ES complex, let's say, binds too strongly, then we're not actually going to get a reaction. We're not going to get any dissociation or any reaction that would lead to the EP. Or if the EP again binds too nicely, it might actually stay there and not dissociate into E and P. And so what they think is that the molecule, when it binds to the enzyme, let's say this is the enzyme and it has a square opening, and this substrate has a circle opening, they're saying that it actually will kind of work its way in there. Let's kind of give it like that. It doesn't want to be a circle. It wants to be a square, right? But the enzyme and the substrate are now bound, and because this is so kind of tight and so kind of uncomfortable, that is what kind of promotes these two things to actually turn into something else and then get out of there. And so some of the lowering of the activation energy is, overcome by forcing these two guys in there where they don't really fit. I don't know. That's where some of the evidence kind of suggests that the induced fit model kind of matches, what we see. Okay. Pause the video. Give these a shot. So the active site is a section of the enzyme. The induced fit model, there's some adapting to the shape. All right, let's do some classification of enzymes. So enzyme names usually end in "ase." They usually describe either the reaction that they do or the substrate that they bind with. This is an example. This is alanine transaminase. It's a aminotransferase. That's the classification. Aminotransferase. So it tells you what it does. It transfers amino groups. Now the actual name of the specific enzyme is alanine transaminase. So we know that it's going to transfer an amino group either to an alanine or from an alanine, but an alanine is going to be one of the substrates. If you think it to the lab that I want you guys to work on for this week, it's amylase. And amylase works on amylose, the component of starch. So ending in "ase" is a sign that it's an enzyme. A couple of differences here. One that tells you about the substrate. So, for example, sucrase reacts with sucrose. It could describe the function of the enzyme. So oxidases do oxidations. It could also be common name. We looked at one just a few minutes ago called chymotrypsin. Pepsin and trypsin are also digestive enzymes like chymotrypsin. They worked a little differently, but they have common names. So these don't end in "ase, but they would be hydrolases, using water to cut, if you were going to classify them. They could also be peptidases. They work on peptide bonds. So there might be some overlap in terms of classifications. There are a lot of different functions and different ways that we could, again, we could say it's a peptidase, we could say that it's an aminase, we can say that it's again, a hydrolase, but let's not get caught up on how many ways can we classify an enzyme, but let's just look at the different classifications for enzymes. So one of the first classification types is the oxidoreductases. So oxidoreductases, says it right in the name, it catalyzes oxidation reduction reactions. This could be an oxidation reaction. This could be a reduction reaction. They could do either of the two, and here are some examples. So an oxidase would catalyze the oxidation of something. We talked about oxidases in the previous example, or in the previous slide. Dehydrogenase would remove or add 2 hydrogen atoms. So it depends on who you're looking at, right? If you're going to take 2 hydrogen atoms off of somebody, you got to put them somewhere. So that's why it says removal or addition. It's done by the same thing. It's just happening to 2 different substrates. One's getting removed from and one's getting added to. But dehydrogenase sort of says it in the name. And then, of course, if you're losing hydrogens or gaining hydrogens, this is a type of oxidation or reduction. So it fits. One of the ones that we're going to look at, one of the important ones that we're going to see in metabolism, is the oxidation and reduction of NAD plus, NADH. So these guys, NAD plus, NADH, are going to gain hydrogens, lose hydrogens, gain hydrogens, lose hydrogens as part of a cycle, ultimately taking those hydrogens and doing something with them so that we can gain energy. But alcohol dehydrogenase would be an enzyme that can take ethanol and turn it into ethanol. So it actually oxidizes the ethanol and reduces that NAD+. Transferases. Very kind of vague there. So a lot of things could be transferases. Kinases are transferases. We talked about kinases not that long ago. Kinases, I said, transfer phosphate groups onto molecules. Transaminases, the ones we just looked at in that other example, the other slide, a couple slides ago. Amino groups from one substrate to another. So alanine transaminase, we saw in that exact example. Here we have an alanine, and it's actually going to transfer from the alanine to something called alpha ketoglutarate. The alanine, when it loses that amino group, is going to be pyruvate, and alpha ketoglutarate becomes glutamate that we know is a neurotransmitter. So this is an important reaction, not just because it generates pyruvate, which will go on to the citric acid cycle, but glutamate is also important neurologically. So transferases move functional groups between two compounds. Hydrolases, these catalyze hydrolysis, so water cutting. So proteases are like the ones that we just saw, chymotrypsin, pepsin, trypsin. These would break hydrolyzed proteins, so they're called proteases. Lipids, if we want to hydrolyze the ester bonds in lipids, those are called lipases. If we want to hydrolyze the phosphodiester bonds in our DNA, sorry, those are called nucleases. Should put my phone on silent. And so hydrolases, overarching category, using water to cut, contains things like proteases, lipases, nucleases, and yeah. So and again, the nucleases, lipases, proteases, you can tell what they do in the name, sort of saying it in the name. All right, liases. Don't confuse this with ligases, which is the sixth one. Liases catalyze the addition or removal of a group without hydrolysis. So removing CO2, removing ammonia, removing water, adding water, but without cutting. So nothing's being cut, or in the sense that we're not using water to cut. So decarboxylases, deaminases, like the alanine transferase, we can also just have a deaminase, one that just takes it off rather than moving it somewhere. Dehydratases remove water. Hydratases add water. So pyruvate here and pyruvate decarboxylate, this is an important reaction. Once pyruvate gets into, we can get pyruvate from a glucose molecule. Pyruvate can actually undergo a reaction here, and we can lose a CO2 molecule and convert pyruvate into a 2-carbon ethanol. So in this case, it removed the carbon dioxide group. It was a decarboxylase. Isomerases just change one isomer into another isomer. So we know that fructose is actually an isomer of glucose. We know that there's some rearrangement that can happen, and this is why sometimes we get false positives with glucose or fructose solutions when it comes to Benedict reagents or when it comes to Sella-Wanoff tests, because they can turn into each other. And so it would be an isomerase that would catalyze that reaction. Epimerases are actually the ones that rearrange carbohydrates, and isomerases would just be rearrangement in general. So this is an isomerase case. We've got malleate turning into fumarate, and all that's changed here is we've got a cis turning into a trans. So isomerases are rearrangers, and then we've got ligases. Now ligases, the key here, ATP energy. And so ligases are also joining, so kind of like taping together. We see a lot of ligases in DNA. When we have DNA replication, we're going to need ligases to piece together little things called Okazaki fragments in the lagging strand. We'll talk about that when we get there. Other little nicks and repair mechanisms in DNA also require ligases to paste together. And again, so joining of two substrates using ATP. So synthetases can catalyze the formation of bonds between two substrates. Carboxylases can add CO2s to a substrate using ATP energy. And so here's an example of that. Pyruvate, in the presence of energy, or when we have energy to put into the system to get ultimately more energy down the road, we can turn pyruvate into oxaloacetate, which enters the citric acid cycle. Pyruvate plus CO2 and ATP. And so the ATP would go in here, it would probably phosphorylate something on this enzyme to cause the enzyme to change its shape in order to do this job, and then something would come later and take that phosphate off. Or the enzyme doesn't get the phosphate group and it gets transferred to some other molecule, but it does cost some energy. And that's what differentiates ligases from other, reactions that link things together, other enzymes that link things together that necessarily may not cause ATP, which is not actually one of the 6 classifications that we're worrying about here. Of course, anytime you take carbon dioxide off or you put a carbon dioxide on, that actually qualifies as one of the ones we looked at earlier, a transferase. So transferases are like ligases in that they can form bonds. A transferase doesn't use energy, whereas a ligase requires ATP. That should help. All right, so I would pause the video, give this a shot. Hopefully you didn't get any of these wrong. An aminase, amino, dehydrogenase, should be removing hydrogen. Isomerase, cis to trans. Synthetase combines 2 molecules. So synthetase would be not unlike a ligase also if it used energy. All right, let's go over some of the factors that affect enzyme activity. Enzymes are very sensitive to their environment. And if you change the environment too much, whether that be the temperature, the pH, or the concentrations of the enzyme and substrate, we're going to see changes in that enzyme activity. So let's take a look first at temperature. So every enzyme is going to have an optimum temperature, a temperature where it performs the fastest that it can perform, meaning the turnover of substrate into product is maximum. For us, in our bodies, it's around 37 degrees Celsius. We tend to see, now if you take a look at this sort of here, we tend to see as temperature increases in general from a low temp, that reaction rate goes up. Our optimum temperature happens to be right here at 37. And then if we start to go beyond that, it starts to go down a little bit to the point where it may actually go all the way down to 0. And when that happens, it's because it denatured. We know from the last chapter that increasing the heat is going to cause those hydrogen bonds to fall apart. And that means secondary structures and tertiary interactions are all going to just unwind, unfold, and that enzyme no longer is going to have that globular shape. It's not going to have an active site. It just won't work. Low temperatures, we see a decrease in activity just because everything's moving slower. The enzyme doesn't denature at low temp. It just moves really slow. The interactions are less often. And so this is typically why we put food in the refrigerator. We want to slow down all the enzymes within the bacteria and all the other microorganisms on our food so that they don't spoil our food as fast. If you go too high, again, denaturing happens and we lose activity. Now, there are organisms that are tuned to living in high temperature. And so, for example, a thermophile is a creature that can live in really, really high temperatures. So these guys would live in 50 degrees Celsius to 120 degrees Celsius, and they would obviously have enzymes that are able to stay folded and function in those conditions. We have heat shock proteins, which come out, they're kind of called chaperones, and if we experience too high a temperature and our proteins start to unwind or unfold, there are specific proteins that start getting made that come out and start stabilizing everything. So some organisms can deal with a change in temp, a high change in temp to some extent, but these organisms here, thermophiles, are specialized for that. And we've actually taken advantage of an enzyme that was actually, or enzymes, I should say, from these kind of thermophiles in something that we do called PCR. We'll talk about PCR when we get to the chapter on DNA replication and that sort of thing. But if you want to take a look at what PCR is, you'll be ahead of the game. pH. We know that pH has a huge effect on the charge of our amino acids, so the R groups of our amino acids, and that will in turn affect the tertiary structure, the folding. And so every enzyme is going to have an optimum pH. This is really just the pH in which natural folding can occur. As you go outside those pH ranges, we're going to see some drop-offs in reaction rate. So in our bodies, that pH happens to be just slightly over 7, where our optimum is. For a lot of our a lot of the enzymes that are in our cells and dissolved in our blood. We do have enzymes that we want to work at different parts of our body. Like, for example, we secrete pepsin into our stomachs to help with digestion. Well, pepsin is a pH of around 2. And so our stomachs are a pH of around 2. So pepsin functions optimally in a pH of 2. And if you were to put pepsin in a pH of 7, it wouldn't fold properly. So different enzymes will have their own different regions and different pH zones that they work in. You would just find this out by kind of studying the environment, doing a pH test, that kind of stuff. But for our bodies, it's safe to say most of our enzymes need to be at that kind of neutral pH. Enzymes in our body have about a 7.4 optimum pH range. Here are some other, so pepsin, you can see here optimum 1.5 to 2. Lactase in our GI tract, where it's still a little acidic, here around 6 and 6 to 7 for amylase. Our mouth contains amylase. Our mouths, probably being closer to the 7 side. There is amylase, again, in our small intestine where it's a little more acidic. Lipase in our pancreas, around 8, and arginase in our liver, 9.7, just to name a few. Different ranges. Enzyme concentration. So the two things that we can talk about as far as concentration goes is enzyme and substrate. So we'll start with enzyme. So enzyme concentration, if you add more enzyme, reaction rate's going to go up. This is the limiting factor. We can only do as much reaction as there are enzymes to do them. So you can very quickly run out of enzymes. You can saturate your enzymes with substrate, and then at that point, the reaction just doesn't go any faster. It's already at its maximum speed. But if you're adding enzyme, more enzyme, more enzyme, more enzyme, you're always going to have a faster rate of reaction. Now, as far as substrate goes, there are kind of two phases. In the beginning of the reaction, where there's no substrate, let's say, there's this little window, let's say from here to here, where we can add substrate, and the reaction rate is going to increase exponentially. This follows what we call second order kinetics. We add some substrate, and it actually doubles or quadruples the rate of the reaction to a point where all of the enzymes are saturated. And at that point, no more substrate that we add is going to affect the rate because, again, we ran out of enzyme. And so if we were also adding enzyme here, well, then this curve would just keep going. As long as we kept adding substrate and enzyme, it would just keep going. Actually, if we keep adding substrate and enzyme, it might seem like it's just going the same rate. If we didn't add more substrate and just added more enzyme, we would see the rate go faster. So yeah, we have to add one or the other. But again, when we add substrate, initially it'll increase the rate of reaction, but eventually we hit that maximum, and then no amount of substrate is going to make a difference. So go ahead and pause, give yourself a shot at these, and then see if you're right. All right, let's talk about how enzymes get regulated. What's shown here is that phosphorylation that we've been talking about, that kinases are able to do, transfer of a phosphate group from one molecule to another. Often in enzyme regulation, phosphorylation is used to turn on an enzyme or to turn off an enzyme. And so let's take a look at that. All right, so regulatory enzymes control the rates of the enzyme catalyzed reactions. They can increase the reaction rate when a particular substance is needed, when concentrations become low, and they can even decrease the reaction rate when the concentration of the product becomes high or when it's no longer needed. And this can be done through allosteric enzymes, feedback control mechanisms, and covalent modifications. To name a few, there's actually quite a bunch of different ways that enzymes can be regulated. Now, allosteric enzymes are those that have regulation at an allosteric site. This is just another site, allo meaning other, than the active site. binding of the allosteric site often causes a shape change. That means the active site also changes in shape and no longer binds the substrate. There can be allosteric enzymes that are positively regulated and negatively regulated. A positive regulator changes the shape of the active site so that it binds substrate more effectively. A negative regulator does the opposite. It changes the shape of the enzyme so that the active site no longer binds effectively. So 2 examples here. So you can see the allosteric site is an other site to the active site, which in this case is the square side of this. In this situation, the top one, binding of a positive allosteric regulator causes a shape change that now better fits the substrate. In the negative case, it causes a shape change which now no longer fits the substrate. So this is regulation through an allosteric enzyme. Not all enzymes are allosteric. The ones that are, that's how they can be regulated, or part of how they can be regulated. There are also some mechanisms that are at work. Here's a feedback control mechanism. This is when too much of a particular product ends up inhibiting its own synthesis. So take a look at this here. So we've got a series of enzymes. Enzyme 1, enzyme 2, enzyme 3. Substrate initially, end product, circle. So what happens is, when there is no end product, then there wouldn't be anything binding here, and so this enzyme would be active, right? The allosteric site would be empty. Substrate would bind it. That enzyme would convert substrate into this green square. Second enzyme would turn it into this rhombus of some sort, and then this would turn it into the circle. Now, as the circle starts to build up, the end product of this reaction builds up, it actually goes and binds to the allosteric site of that original enzyme, the first enzyme, inactivating it. And by binding to it, it changes the shape of the active site, so substrate no longer binds. And so this is a negative feedback control, where the product at the end of the sequence inhibits the early enzyme. This is how your body keeps from making a bunch of stuff it doesn't need. And then as the end product gets used up, whatever it is that its job is, it becomes less and less prevalent, and it's not bound as often to the enzyme. And so then we would have unbound enzyme ready to make more of that product again. Covalent modification. This is making covalent bonds to enzymes. Some covalent modification is reversible. Others we'll see when we look at how to inactivate enzymes can be irreversible. Modified when covalent bonds to a group on the polypeptide chain are formed or broken. So covalent modification could be like adding a methyl group. It could be like taking off a methyl group. Zymogens, or proenzymes. So these are 2 terms that basically mean the same thing. These are inactive forms of enzymes that can be modified later to be active. Chymotrypsin, we talked about that. It was a digestive enzyme. This one actually is made in one part of the body, but used in a different part of the body. And you would expect that with a digestive enzyme. We don't want it to be digesting things, as soon as it gets synthesized. So it's made as a proenzyme that requires certain modifications, in this case, certain cleavages in its peptide chain that let it fold into a new form, and then it's an active enzyme. So that's what a zymogen is. Now, lots of zymogens are digestive enzymes, protein hormones, blood clotting enzymes. Let's see, proteases, or digestive enzymes that hydrolyze proteins, like chymotrypsinogen is the zymogen, and chymotrypsin is the active enzyme. Anyways, you can see that there's quite a couple of examples here. Insulin made as proinsulin, fibrin, which helps with the clotting from fibrinogen. Okay. Actually, this is that exact example I was mentioning. So your book has this in here. So chymotrypsin is formed first as chymotrypsinogen. And until it gets these two little sections, these dipeptides cut out or excised, then it's in its inactive form. Once those are cut out, it's in its active form. Phosphorylation. So again, kinases can activate an enzyme or deactivate it. And here we see an inactive enzyme. A kinase comes in here, uses ATP energy to add a phosphate. That's where those phosphates usually come from. This is why we have ATP. This is the actual usage of that energy currency. And so now this enzyme is turned on by the phosphate group and active. This is an inactive enzyme that has a phosphate group. We come in here, phosphatase takes it off. Now it's an active enzyme. So phosphorylation is just another way of turning on or turning off the activity of these enzymes. All right, pause the video, give this a shot. A phosphorylase is a covalent modification. So this is kind of a general term. Phosphorylase, adding a phosphate group, that counts as a covalent modification. So phosphorylation is just a subset of covalent modification. Things like iodination or methylation, hydroxylation, adding a hydroxy group, a CH3, right, and iodine. One of the neat things about enzymes is that different parts of the body actually exhibit slightly different forms of the enzyme. We call this isoenzymes. So isomers of the enzyme, slight variations due to the amino acids. So for example, there are 5 isoenzymes of lactate dehydrogenase. And doctors are able to use which particular isoenzyme they find to indicate what parts of the body perhaps might have some damage. So this comes from the fact that when you experience myocardial infarction, there are a couple of increases that can be monitored, like the levels of creatine kinase and lactate dehydrogenase. Now for lactate dehydrogenase, the fact that different forms exist in different tissues allow them to kind of diagnose different damage to disease, different damage or disease to a particular organ. And so in the heart, we're going to see this particular isozyme that has 4 of these H subunits versus in the brain, let's say, we're going to see, or in the brain and lungs, we might see this particular isozyme with 2 of the H and 2 of the M. Or in the liver, we would find only the M portion. These are different subunits, so these would be different quaternary structures. So this just happens to have 4 of the M version of this particular enzyme subunit. So anyways, this is neat. This is useful information that scientists can use to diagnose. Okay, let's talk about inhibition. Inhibition can take place a few different ways, and it can involve actual binding to the active site or to the allosteric site. It can be reversible, it can be irreversible. So as far as how we inhibit, we call these inhibitors. They're molecules that, as it says, or as you would imply, reduce the rate of the reaction. They can prevent the substrates from binding into the active sites directly or indirectly through changes to the quaternary structure of the protein. And it could be reversible or irreversible depending on the nature. We'll do reversible first. Reversible just means that when you get rid of the inhibitor, the enzyme goes back to normal. The key feature here is that there are not covalent bonds formed. covalent bonds are going to be key in our irreversible. Reversible can be competitive or non-competitive. Competitive just means that it's going to compete for the active site. So where substrate is normally trying to bind, competitive inhibitors kind of interfere. Non-competitive means that that's not the case. They go for a site other than the active site. A competitive inhibitor probably looks like the substrate. It's going to have similar structure and polarity. It's got to recognize the same spot on the enzyme, so it's probably going to look a lot like the normal substrate. You can reverse the effects of this inhibitor by increasing the substrate concentration. It'll overwhelm, outcompete. We get back to normal reaction levels by increasing substrate concentration. An example of a competitive inhibitor are these antimetabolites that are actually used in bacterial infections. The inhibitor looks an awful lot like the normal substrate. So you can see here the only difference is there's a missing CH2 group somewhere in here. So it still recognizes the same spot on that in that enzyme, and it just knocks it out, binds there, outcompetes it with the substrate. Eventually the bacteria will die, because it's not getting enough of whatever product was supposed to be made. Another one here, sulfanilamide competes with p-aminobenzoic acid. Similar structures are a little bit different in this region. So you might think maybe this is the binding region. Could be. Non-competitive inhibitors, these go for the allosteric site, and by binding to the allosteric site, they distort the shape of the enzyme and they prevent binding of the substrate. This cannot be reversed by adding more substrate. Since substrate here doesn't matter, substrate is free to bind to the active site if it can. Adding more isn't going to change the fact that some of your enzymes have bound to inhibitor. So this is not reversed that way. The only way to reverse this is to maybe do a chemical reaction that's going to come in here and kind of scoop out all the inhibitor, just kind of remove it from the reaction so that the substrate is available to go into the active site. Now, as far as irreversible inhibition, enzyme activity is destroyed because we end up getting some kind of covalent bond. So inhibitor covalently bonds with either the R groups of amino acids that are near the active site, or it changes the shape of the enzyme, permanently preventing the substrate from binding. And so here's an example of that. We've got this DFP is a inhibitor. This is the presumably inside the active site of this enzyme. This is on the serine residue, this OH, and so this binds, reacts with that OH, forming a new bond to this inhibitor. Now, of course, if this is in the active site and something wanted to come in here and interact with that OH is gone, and this enzyme is no longer going to be able to react with that substrate. These are some irreversible enzyme inhibitors. So cyanide, this is why cyanide's a poison. It'll bind actually to the iron in your heme group and inhibit either electron transport. It doesn't say anything about oxygen binding, but if it likes iron, it could possibly do that as well. Penicillin is an irreversible inhibitor for bacteria. It inhibits the enzyme that builds the cell wall. And yeah, so those are some examples of irreversible inhibitors, and this is a summary of those details. All right, give these a shot. Find out or decide if this inhibitor is competitive or non-competitive. All right, hopefully you were able to get that correctly. All right, let's conclude our chapter with the discussion of vitamins and coenzymes. Some vitamins are coenzymes. We'll talk about what coenzymes are in a second. Organic molecules, vitamins, organic molecules that are essential for normal health and growth, required in trace amounts. Some need to be obtained from our diet, others we can kind of synthesize on our own. most of them probably, well, anything that we talk about is stuff we get from our diets. If we make it ourselves, we tend not to worry about it. Are grouped into water-soluble and fat-soluble. So the water-soluble vitamins are the ones that can be cofactors, and this is because cofactors need to be able to dissolve in the cell, just like the enzymes, and so fat-soluble vitamins wouldn't be floating around useful in that way. So the water-soluble ones are often the cofactors, or in some cases precursors of cofactors. So because they're water-soluble, we can't store them, so we need to get them from our food each day. They get excreted in urine. They're destroyed by heat, oxygen, ultraviolet light. So often in food preparation, the vitamins kind of get destroyed in the form anyways that we want them. So you really want to be eating fresh, fresh fruits and vegetables to be getting those vitamins. And then again, they are cofactors or precursors of cofactors. These are things like vitamin B1, vitamin B2, vitamin B3. You'll notice I'm not going to go into any details here. You can read for yourself the examples of the enzymes that they work in and the kinds of ailments you get for being deficient in them. Again, the deficiency leads or comes from the fact that the enzymes that require them aren't able to do their jobs, and so then those products that would normally be made aren't getting made. Vitamin B3, vitamin B5, vitamin B6, most of the B vitamins, B12, vitamin C, vitamin H. I think there was one more vitamin there in the beginning. Was it vitamin A? It wasn't A. Anyways, these are the water-soluble vitamins. Now, the things you should be working on or focusing on is how to tell if an enzyme is a functioning enzyme with or without a cofactor. And so these are good questions for that. If it requires a metal ion, and they've changed the definitions recently, but there used to be two terms. It was cofactor and coenzyme. And the coenzyme were small organic, actually, that's wrong. The cofactors were small organic or metal ions, and the coenzymes were like vitamins or larger organic molecules. So requiring magnesium means that it needs a cofactor. That's a cofactor. Nowadays, they just call everything a cofactor or a coenzyme. They're interchangeable, and they don't mean specific things. Requires vitamin B. That would be obviously an enzyme that requires a cofactor. Requiring magnesium is a cofactor. An enzyme that has 4 polypeptide subunits. Doesn't say anything about it needing, another component. So that would be a simple enzyme. Okay, fat-soluble vitamins. These, you should just know we can store them, so they're not so much important that we get them in our diet. These are not catalytic. They don't do anything in enzymes. They don't have any role in those reactions. Vitamin A is going to be responsible as a precursor for a molecule retinol in our vision. Vitamin D regulates a lot of the calcium uptake in our bodies to help with bones. Vitamin E is an antioxidant, so it's important for scooping up electrons from free radicals so that they don't go and destroy various components, important things like our DNA or our molecules that we need. And vitamin K has a role in blood clotting as precursors to some of the clotting factors. So you can look it over some of those. Here's retinol. Oh, sorry, here's vitamin A, precursor to retinol. Vitamin D. We actually synthesize vitamin D inside our skin when we get some UV light from the sun. So if you go out in the sun, you should have plenty of vitamin D. If you don't, milk is fortified with vitamin D so that you can get that vitamin. Here's vitamin E, and again, It's an antioxidant. That means that it's a kind of a sacrificial molecule. If something is going to get oxidized, it's better for this to get oxidized than something more important. It also has a role in avoiding hemolysis and anemia. And vitamin K here, needed for the synthesis of zymogens for blood clotting. So zymogens, remember, are precursor proteins, precursor enzymes, having to do here with blood clotting. All right. Pause this, go back and look at which is which and test your knowledge. Okay. Identify the vitamin associated with each of the following. Again, you can, don't spend too much time memorizing what each of these do. I'm not going to test you really on that. I mean, I didn't in the past. Now that you can probably look this up, while you're doing your test, I may or may not give you a question on a vitamin and specifically what it does. But in the past, where this was sort of just information really that I was focusing more on just the water soluble, water insoluble, or fat soluble types, of knowing which belongs to which category, which vitamin belongs to which category. But I suppose now, since it's a kind of an open book test, I may ask something like this. All right, so that's the end of the chapter. So enzymes, proteins that have tertiary or quaternary structures that have active sites, bind substrates, form ES complexes, EP complexes, and make products. Their activity is affected by changes in pH, temperature, concentration, inhibitors. It's also controlled by allosteric enzymes, feedback control, zymogens, covalent modification, and then may need cofactors, which could be metal ions, or what has been called coenzymes, the vitamins, which can be water soluble or fat soluble, but we're not going to say that fat soluble are things that they may need, because we know that they can't use them, but vitamins can be both. All right, everybody, so that's the end of this chapter, and like I said, I'll be trying to get a quiz up for you guys this weekend before spring break, and another lecture and quiz up by the next weekend, hopefully. Quiz-wise by the next weekend, and lecture-wise by this weekend, so that next week during spring break, if you guys are going stir crazy, you have something to do. All right, take care, everybody.